forgot to hit record before I started my spiel. Fine. So about a year or so after we started the Peggy Lillis Foundation, I apologize for the passing noise here. Work from home. So about a year after we started the Peggy Lillis Foundation, we started seeing a lot of information about fecal transplants as a treatment for people that had recurrent C. diff. And so it was kind of too late for my mom. There definitely were a few doctors that had started doing them at that point, but it wasn't something that was sort of pretty well known. And so as we've grown over the years and become more and more involved in public policy and advocacy, the Food and Drug Administration is sort of a clear avenue of things that we want to look at. And so as FMTs become more widely available, we have had people come to us, including advocates and people who are seeking answers, and say, like, well, why is fecal transplant regulated the way it is? Why can't I just get it? Like, why does it...
Be in FL, Minnesota.
Hi, if you're on the phone, please mute yourselves because we can hear you. Thank you. So, anyway, that's throwing me off for a second, but anyway, the good stuff. So in Professor Hoffmann is going to get into some much greater detail, but basically in 2013, the FDA began to issue its regulations on how to regulate fecal microbiota transplant. There's been a lot of changes, but there's also been a lack of, I would say, transparency in terms of patients, because their public meetings are all in DC, and if you're, can be very difficult to get to, but also a sort of lack of just making a sort of decision. And so that's left both people who are providing fecal transplants as a treatment and also companies that are developing microbiome-based solutions in kind of a no man's land where we're not entirely sure how this is going to work out. So I'm really, really happy. This is the first of what are going to be quarterly webinars that the foundation is going to be doing this year on different subjects. We really want to educate our supporters and stakeholders on ways that we can all collaborate and work together. And so to that end, we are delighted to have Diane Hoffmann to present us, to do a presentation on understanding the regulation of FMT, past, present, and future. I'm going to just give a little bio intro here. So Professor Hoffmann has been on the faculty of the University of Maryland School of Law since 1987. She's taught torts, law and medicine, healthcare law, legal issues of the elderly. Her research includes the intersection of law, healthcare, ethics, and public policy. And she was a primary author of Maryland's Healthcare Decisions Act, dealing with advanced directives, surrogate decision-making, and guardianship for individuals lacking healthcare decision-making capacity. Most of the times that I've gone to these FDA meetings, Professor Hoffmann has been there and spoke, and I've been really impressed by her breadth of knowledge and also her sort of the way in which she sort of takes everybody's, all stakeholders' needs into account. So Professor Hoffmann is going to speak for about 40 minutes, and then we'll have some time for questions and answers, and we will let you know how to do that when we get to that point. So I'm going to turn it over to Professor Hoffmann. Thank you so much. And again, if you can, please mute your phones or mute your audio so that we don't have background noise while the professor's talking. Thank you, Professor Hoffmann.
Thank you, Christian. It's a pleasure to do this, and I'm really feel very fortunate to be able to work with groups like the Peggy Lillis Foundation. So as Christian said, I'm going to be talking about, he asked me to speak about understanding the regulation of microbiota transplants, and I thought maybe the best way to approach that was by talking about the past, how we, the past and history of fecal microbiota transplants, as well as their regulation, and then what's happening now and what might happen in the future. So just a little bit of history. Many of you may already know this, but the practice of FMT was described back in the 4th century by a Chinese researcher by the name of Ji Hong, who first described it to treat patients who had diarrhea or food poisoning. And then jump forward a number of centuries. In the 16th century in China, there was a doctor, Li Shizhen, who gave it orally as treatment for severe diarrhea and called it a yellow soup. Then as early as the 17th century, veterinarians recognized the value of FMT. They were using it to treat horses with diarrhea. And in World War II, camel stool was used by German soldiers in North Africa to treat bacterial dysentery. But it was in modern medicine, the first published research on the concept of FMT was by Ben Eisman and colleagues in 1958, and that involved fecal enemas as treatment for antibiotic-associated diarrhea. In 1958 to the early 1960s, there was a brief uptake of fecal enema treatments in hospitals, but also around 1958, vancomycin was approved by FDA, and it reduced the need for FMT using fresh stool from donors. And then between 1958 and 2010, there were almost no reports published on the technique in the medical literature, very few. And physicians became to rely more and more on broad-spectrum antibiotics, which led to the rise of multi-drug resistant organisms. Then over the past decade, FMT became more widely used due to a rise in the incidence, the morbidity, and mortality associated with recurrent C. difficile. And new strains of C. diff became more infectious and virulent at the same time as doctors were prescribing more broad-spectrum and potent antibiotics. Some strains of C. diff have become resistant to the available antibiotics. So then jumping ahead to the use of FMT to treat recurrent C. diff, the first documented case was in about 1983. And in terms of the clinical application of FMT, it wasn't until the last 7 or 8 years that FMT came into mainstream medical use for recurrent C. diff. The procedure is generally performed by a gastroenterologist and calls for fresh or frozen stool product from a healthy donor, and it's diluted with a liquid, usually saline, and then delivered into the intestinal tract of another individual. And the procedure can be performed by enema, colonoscopy, sigmoidoscopy, or a nasogastric tube. And in some cases, the stool product is encapsulated and taken orally. The first physicians performing FMT in more recent times, have basically obtained the stool used for the procedure from friends and relatives of the patient. And they had to both screen the donor and prepare the stool for administration themselves, which was somewhat time-consuming and not a particularly pleasant process, although not all that difficult in terms of equipment and know-how. Then, in 2012, an MIT postdoctoral student opened a nonprofit stool bank called OpenBiome outside of Boston, Massachusetts. It was the first freestanding stool bank in the country. The stool was from healthy donors who were often students at Tufts University, and that was simply because OpenBiome's office was near Tufts. And college students are generally young and healthy. Each donor had to complete a 120-question survey about their travel history. With a physician, they had to disclose recent antibiotic use. If the potential donor passed this initial screening, A sample of their stool was then reviewed by a lab for any infectious agents, as well as to determine the health of the bacteria. The donor's blood was also tested for standard bloodborne diseases, as well as hepatitis A, B, and C, syphilis, and HIV or AIDS. Each donor is paid about $40 per sample. I believe that's still the case today. You're paid $40 a sample for your donation, but there's even a more extensive questionnaire today, and there's also testing for several antibiotic-resistant bacteria. Now, OpenBiome currently provides stool for almost 11,000 FMTs per year. Other FMTs are performed using stool from hospital stool banks, so there are also a number of hospital stool banks, usually associated with medical centers, academic medical centers. Also, the literature indicates that many FMTs are performed by individuals at home, the do-it-yourself method, without the benefit of screening and administration by a healthcare provider. There are a number of websites that provide information about how to do the procedure with minimal equipment or resources. And I think the reasons for do-it-yourself option may include the inability to find a physician who will do the procedure, at least initially. I think that's changed quite a bit. The ability to obtain stool from a friend or relative, the cost of going to a physician or medical center, and lack of insurance coverage for the procedure. Based on the evidence to date, though, on safety and efficacy, the procedure has become the standard of care for recurrent C. diff that's unresponsive to traditional antibiotic treatment, and it is covered by a number of insurance companies. While a few physicians were performing FMTs before 2010, the legal status of the procedure at that time wasn't really clear. A handful of physicians had made inquiries to FDA as early as 2010 as to whether they needed an investigational new drug application for research on FMT or to treat patients with C. diff or other serious gastrointestinal conditions. Now, FDA responded to those inquiries, but it was on an individual case-by-case basis. They made no public announcement of how they intended to regulate the procedure at that time. Then in 2011, RebuyOtix, a company that planned to market a stool-based product, specifically a cryopreserved filtered microbiota derived from the stool of screened donors and administered through an off-the-shelf enema delivery system, was formed. In 2012, RebuyOtix representatives met with staff at FDA to determine whether it would need to go through the investigation.
Okay, I'm listening now to something. I'll come by later.
Application to market the product.
I think it's over at 3 o'clock.
The company...
Hey guys, Professor Hoffmann, wait a second. If you are listening in through your phone or your computer, please mute it because we can hear people answering their phones. Thank you. Thank you.
So RebuyOtix met with FDA and asked them if they needed an investigational new drug application to market the product. And I'll say a little bit more about that in a bit. That would mean it was a drug and that would be regulated as drug. At the same time, they also requested a determination from the tissue reference group at FDA and asked for a determination as to whether the product could be treated under regulations for human tissues and cells. But FDA, or the tissue reference group, said that human stool is not human tissue. Rather, it's made up of live microorganisms, so it didn't meet the definition of a tissue. And then shortly after that, FDA told RebuyOtix that its product would require an IND and would have to go through the necessary clinical trials for new drug approval. So just a little bit more background on what the implications of FDA's decision for RebuyOtix meant. it would be regulated as a drug. Stool would be regulated as a drug, their product. That meant that it would have to establish safety and effectiveness, and would have to file an investigational new drug application. And that involves pharmacological and toxicological evaluations, chemistry and pharmaceutical manufacturing and controls data, animal testing, a clinical plan for the development of the new drug product, a specific protocol for the initial clinical trial, and then it has to go through 3 phases of clinical trials generally. The first phase is with a relatively small number of healthy individuals to just basically test for safety. The second phase includes more individuals and is for both safety and effectiveness, starting to give it to people who have the condition that you're hoping to treat. And then the third phase, hundreds more patients are in that trial, and it really focuses on efficacy. And these are generally randomized controlled trials where you have one group receiving a placebo or an alternative drug and one group receiving the tested drug. In this case, it would be stool, and they're randomized into different groups. And the literature indicates that now it costs about $1 billion at least to go through the drug approval process, and takes about 10 years. This is on average. So obviously it's a very costly and lengthy process. In contrast, the regulations for, if we were going to regulate stool as a tissue, it's a very different type of regulatory approach. It's some of you may be more familiar with blood donation. It's what is more likely done when you have different donors and you're giving their blood or tissue to a recipient. And the primary focus is on prevention of communicable disease and transmission. There are 2 types of tissue. There are some that are just regulated as tissue, and there are others that are regulated as both a tissue and a drug. But if it's regulated as, well, either way, as a tissue, there are a number of regulatory components. So the manufacturer would have to register the facility and all the products with FDA so that they know that someone is producing this, and that allows them to inspect and enforce any kind of compliance issues. They have to have in place relevant donor screening and testing, as well as current good tissue practices. And good tissue practice refers to how you're going to recover, process, store, label, package, and distribute your product. The manufacturers also have to track each cell and tissue product so that in case of an adverse event, the root cause may be investigated. And as I said, a tissue product that meets the criteria for regulation solely as a tissue is not subject to pre-market approval by FDA, but tissue products that do not meet the criterion for regulation solely as a tissue usually have characteristics of a drug and must also comply with the drug requirements for pre-market approval. So as I left off with what FDA was making individualized case-by-case decisions up until May of 2013, when the agency announced that it would classify stool as both a drug and a biologic at a public meeting. So a drug is any article that's intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animals. A biologic is a subset of a drug. So biologic is also a drug, but it's a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component or derivative, allergenic product, protein, or analogous product applicable to the prevention, treatment, or cure of a disease or condition of human beings. So by classifying FMT as a drug, it resulted in what I've heard was less than 20 physicians in the country being allowed to perform fecal transplants. So in response to this ruling, there was a groundswell of opposition from physicians and patients. And just 2 months later, in July of 2013, the FDA kind of stepped back from its initial position and announced that although it was still going to classify stool as a drug and a biologic, it would exercise what it calls enforcement discretion and not enforce the IND requirement for physicians who were performing FMT on patients with C. diff that was unresponsive to standard therapies, and that an IND would, though, be required for any other indication. At the time, FDA also stated that it intended to exercise its enforcement discretion on an interim basis while the agency further considered the matter. It was the agency's decision that it would exercise its enforcement discretion that paved the way, though, for more widespread use of FMT by physicians for the treatment of recurrent C. diff. But then, in March of 2014, FDA published draft guidance that would add another qualification to the use of FMT for C. diff. And that was that the FMT product must be obtained from a donor who is known to either the patient or to the licensed healthcare provider treating the patient. Although the 2014 guidance did not mention stool banks, it would have effectively foreclosed their use. Until the guidance became final, though, FDA indicated that it would continue to exercise its enforcement discretion and allow physicians to obtain stool from stool banks without an IND. FDA received many public comments on this draft guidance in favor of allowing patients access to FMT to treat C. diff, including access to FMT produced from stool banks. Despite these comments, though, in March of 2016, FDA issued a revised draft industry guidance clarifying the prior guidance and stating specifically that centralized manufacturing in stool banks presented safety concerns related to the use of FMT from a limited number of donors administered to multiple patients, and that these safety concerns included transmission of infectious agents and potentially other unidentified risks related to changes in the microbiome. Thus, it would not exercise enforcement discretion for stool obtained from stool banks, and that stool banks would have to obtain an IND. But again, this was draft, and so FDA was asked for public comments on the revised draft guidance. I should step back and say, so most people are probably familiar with statutes and regulations as law, as legal rules and standards. Here, FDA does a lot of kind of rulemaking, but it's not really law, through these industry guidance documents. I think they do it when they're somewhat unsure of how to proceed. They can be more flexible by administering this way. On the other hand, industry, I think, is concerned when FDA regulates this way because industry is obviously looking for some certainty, and they can't necessarily rely on these industry guidance documents. But in terms of the comments that FDA received on the 2016 guidance, they received comments from both consumers and patients as well as healthcare providers. So I think it received about 55 comments from consumers and patients who, I think all of them criticized the guidance for placing barriers to patients to FMT access. Many said they feared lack of access to a life-saving therapy and believed that if an IND were to be required for stool banks, they would have to meet stringent trial eligibility standards to receive an FMT and might receive a placebo rather than the therapeutic intervention. These commenters urge the FDA to continue to allow stool banks to provide FMT for treatment of C. diff not responsive to standard therapies. FDA also got about 44 comments from healthcare providers And some of the things they said were that, and they felt that stool banks provided a safe, rigorously tested product in a timely manner, and that in contrast, hospital and local laboratories, especially in rural areas, did not have the facilities or training to conduct the same type of screening as stool banks, and they're often unable to screen donors quickly. Also, they said that screening is expensive and it's not reimbursed by all payers, and it's hardest on poor patients who cannot afford the out-of-pocket costs. So at this time, what's happening is that FDA is continuing to treat stool as a biologic and a drug, but it's still exercising its enforcement discretion so that stool banks do not require an IND. It's clear, though, from everything that I have seen from FDA that they would really like to require stool banks to obtain an IND, but I think they also do want to be responsive to patients and their need for the procedure, at least until a new drug is developed. And I think the future is somewhat complicated for them by the fact that a number of companies are going through clinical trials right now to attempt to develop a stool-based drug that would be approved as a drug. And I think it's important to understand the types of drugs that are in the pipeline. So one way I think of these are on a spectrum as between the natural products. Say you got stool from a friend or family member. That's the natural product. And goes through, maybe some saline is added to it in order to administer it. And then the companies up top here who are going through clinical trials, RebuyOtix, Finch, Series, and Vedanta, are on different places on the spectrum. So RebuyOtix and Finch, their product is much more closer to the natural product. It consists of a community of microorganisms. So stool has many microorganisms. We don't quite understand how they interact or work together, but there's a probability that that's going on, and still uncertain what contributes to the therapeutic benefit. But and the companies on the left side of the spectrum, they retain the donor differences. So there are different donors, and of course, donations differ even when they're from the same donor over time because of what they eat or any medications they take or things like that. So that creates some challenges for when you want consistency for a new drug. Vedanta is the one on the other end, and these drugs are more selected organisms. They take some of the organisms out of the community. They're extremely well characterized, cultivated microbial strains. And then in the middle, where Series is, they have used only a certain fraction of fecal microbiota from individual donors. And they use the bacterial spore fraction to constitute the product. But it also, as I said, retains this donor and donation lot variability. And this chart just shows you these four different companies, sponsors, the stage of their clinical trials. So some are in phase 2, some are in phase 3, the number of participants they have enrolled in the clinical trial. All the clinical trials are multi-center, double-blind, randomized, placebo-controlled trials. And then on the right hand, it shows you what the formulation of the product is. So there are challenges of the drug biological regulatory framework for those products that are on the left-hand side of the spectrum. They don't fit the typical product that goes through the drug approval process. And some of those challenges, as I've already mentioned, such as the donor and sample variability, because you want consistency for a drug. There's limited data on pharmacokinetics, how the drug works in the body, and pharmacodynamics, what are the interactions between the drug, the human body, and the pathogen that might be causing the infection in the patient. Also, preclinical animal models provide very limited usefulness. And these things are generally required in all IND submissions. But I think the FDA is making some exceptions here just because they would have to for the product to go through this process. Also, there are no diagnostic tests based on metagenomics characterization of the donor microbiome, and that could, for example, identify antibiotic-resistant genes. So that's if you have we have some tests now that do test donors for them, but there is not a general, I guess, metagenomics characterization that can be done for that. Also, the framework doesn't easily incorporate new tests and technologies, like, for example, is done in transfusion medicine. So think about blood donors certainly have to get certain tests to make sure that they're not carrying certain blood-borne diseases. But if a new test, or there's a new virus, for example, and a new test is developed to test for that virus, we can simply test new donors going forward. It's not difficult to incorporate that new test in the process. But what if you have a product already on the market that has been developed through stool from individual donors, and you discover a new antibiotic-resistant bacteria, and that some of your donors had that? You have to take all those products off the market. So it creates some difficult challenges. Also, another challenge is ensuring product potency. The current approach is simply to define a dose in terms of viable bacteria. And there may be other metrics in the future, but things maybe like the diversity of the microbial community, but we're not there yet. Another more legal-based challenge, I think, or problem for having something go through the drug approval process is that newly approved drugs would be entitled to off-label use by physicians. Off-label use allows physicians under what's called the practice of medicine, which is not regulated by FDA, to use the approved product for other indications that FDA has not approved. An example is treating certain types of pain with tricyclic antidepressants. And I think there's a real question about whether we should allow off-label use of stool-based drugs. FDA has not permitted FMT for indications other than recurrent C. diff because of lack of data on effectiveness. If a new stool-based drug were to be approved, that under traditional drug law, off-label use would be permitted. So despite the problems with this drug model, I think FDA does not seem to want to change the regulatory paradigm that they're using. They'd rather keep with the drug biologic model and enforcement discretion, at least for now. However, the number of the companies that are going through the clinical trial process, RebuyOtix, Series, and Vedanta, have raised tens of millions of dollars from investors. There's an article in the New York Times that I'm referring to that talked about this. About a year ago, they formed an association to advance their interests with the FDA. I think they would like the FDA to stop the enforcement discretion practice, as they argue that they're having a very difficult time getting subjects to enroll in their clinical trials. My own view, and I have no real insider information about what FDA is going to do, but I think it's unlikely that it would stop the enforcement discretion until a new drug is approved. but I could be wrong. But if I'm not, I think still a big question is, once a new drug is approved, what will FDA do? Will it let the stool from OpenBiome, for example, stay open, and OpenBiome continue to be able to provide stool? One way that FDA could deal with this is like they do for other biologics and orphan drugs. So new biologics may be able to claim something called data exclusivity, and if they're designated as orphan drugs, they may be able to claim market exclusivity. So what does that mean? So biologics, which is what any of these drugs that are in the pipeline would be, would get 12 years of data exclusivity if they were approved by FDA. And basically that protects approved biologics from competition by products for the same indication that are highly similar to the reference product, which is the first approved product, as long as it has no clinically meaningful differences. So before 12 years, a product could try to go through the drug approval process, but they'd have to start from the beginning and do all the clinical trials. After 12 years, that second product could refer to the data from the reference drug, and they wouldn't have to repeat the same clinical trials and research before going to market. So if we assume that the OpenBiome product was the second in line and one of these other drugs got approved, I think a question is the stool product used by the stool bank highly similar to and have no clinically meaningful differences from the approved biologic? And if it were something, it probably depends on whether it's one of the products that were closer to the natural product, like the RebuyOtix product. One thing that is different that FDA takes into account is the method of administration. So the RebuyOtix product is administered by enema, whereas the OpenBiome product is administered generally by colonoscopy, or I think orally as well. They have capsules. The other issue is that basically all of these products have been designated orphan drugs, and the orphan drug designation carries with it market exclusivity for a period of 7 years. So an orphan drug is defined as one that would be used to treat less than 200,000 people per year. And now, although the incidence of C. diff is about 500,000 per year, only about 100 to 150,000 develop recurrence. So because the drug they're developing is focusing on recurrence, it would meet the definition of an orphan drug. But if one of those drugs is approved by FDA, another company can't market that same drug for the same orphan drug indication during the period of market exclusivity. And so the same drug is defined above on the slide, that one that contains the same principal molecular structural features and is intended for the same use as a previously approved drug, except that if the subsequent drug can be shown to be clinically superior, it will not be considered to be the same drug. So in terms of how that might apply to OpenBiome, OpenBiome, again, isn't really going through the drug approval process, but in terms, again, if it were, we'd look at it and how it compared to the newly approved drug. And again, if it's RebuyOtix, I would think it might be the same drug. If it was Series or Vedanta, probably not. If it was determined to be the same product, then we'd have to look at whether or not it was clinically superior, which means that the drug has a significant therapeutic advantage over and above that provided by an approved drug, because it has greater effectiveness, greater safety, or otherwise makes a major contribution to patient care. Thus far, FMT with donor stool has not been compared in a head-to-head trial with the RebuyOtix or Series products. But there is a good possibility that FMT would be found to be superior in terms of effectiveness. That's a possibility. In addition, or alternatively, OpenBiome advocates could argue that their product makes a major contribution to patient care, referencing the criteria for clinically superior. So in its efforts to try to figure out what to do going forward, just last year, November 4th, FDA held a public hearing to obtain public input on the state of the science regarding FMT to treat C. difficile infection that's not responsive to standard therapy. They sought comments on the clinical evidence of effectiveness of FMT, safety information, the impact of FDA's current enforcement policy on FMT product development, and a future path forward. Both Christian and I testified at this hearing. I chose to comment on the last two items. And in terms of enforcement discretion, I think it's fair to say that there have been both pros and cons. I think largely pros. It has had, on the one hand, I think the relatively light regulatory burden for conducting research under the enforcement discretion regime facilitated a very rapid transition from what used to be a crude procedure that involved preparation and administration of raw homogenized stools to easily administered, purified, cryopreserved microbiota that's centrally manufactured from rigorously tested donors. It's also enabled investigators to perform a number of studies comparing fresh and frozen thawed microbiota, routes of administration, and even dose assessment and treatment of recurrent C. diff. Investigators have also been able to make preliminary assessments of the clinical safety and efficacy of FMT in different higher risk recurrent C. diff patient groups. So people with inflammatory bowel disease or advanced liver disease and organ transplant recipients. And these higher risk patients are disproportionately represented within the recurrent C. diff population, but they're excluded from in the formal clinical trials conducted by commercial developers under their investigational new drug clinical trial protocols. Another big pro is that it has permitted access to the procedure for thousands of patients suffering from C. diff, recurrent C. diff, who may not otherwise have been able to obtain relief. But there's also been one significant disadvantage, and that's been that it hasn't been accompanied by data collection, which has been an enormous missed opportunity to collect clinical outcome data from tens of thousands of patients. In terms of reasons to continue the enforcement discretion or something that will allow continued access to patients, if FDA ceases its exercise of enforcement discretion, OpenBiome, I think, could close. And one of the immediate impacts, now I'm assuming this is after a new drug is developed, one of the immediate impacts on patients would likely be the cost of a new drug compared to FMT. FDA approval of FMT products is likely to lead insurance companies to provide coverage for them. However, if the cost is substantial, it's also likely that insurers will limit coverage and consumers will be required to pay a significant amount out of pocket. All these products are biologics, and they are among the most expensive on the market. And the increase in cost for patients associated with one of the new FDA-approved drugs may push some patients to attempt to find a physician who will perform an FMT with donor stool from a family member. But physicians may not want to do this if they don't have access to the stool from a stool bank. They may prefer not to have to prepare it themselves. And also, if that's the case, then we could see an increase in the number of do-it-yourself efforts, which has a number of risks from inadequate donor screening and testing for infectious diseases and microorganisms. So is there another option? Well, one thing that a colleague of mine, Alex Koretz, have written about is the possibility of allowing OpenBiome to operate under a permanent IND or ongoing IND. It's unlikely that if FDA terminates its enforcement discretion policy for stool banks, OpenBiome, for example, wouldn't pursue a traditional IND for approval as a biologic because it's not for profit and it would have to raise significant investment funds to do so, and investors would want to work with a for-profit entity. So a requirement that stool banks obtain an IND in order to provide their stool product to physicians for recurrent C. diff could potentially be addressed by what I said, an ongoing observational clinical trial that engages in data collection on safety and effectiveness, but that need not make progress toward obtaining a new drug approval or a biological license. This kind of trial could be conducted under a research IND. It would be an observational study in which the patients would not have to be assigned to control and intervention groups, and so it would differ from a randomized controlled trial. The stool bank would sponsor the IND and would be responsible for submitting the IND application and enrolling physicians into the study, would also be compiling the safety and efficacy and adverse event data, and then reporting it back to FDA through safety reports and annual reports. And the inclusion criteria for participation in the trial could be very broad, permitting basically any patient with recurrent C. diff for whom a treating physician believes an FMT would be beneficial to enroll. And then physicians participating in such a trial would be required to report adverse events and safety and effectiveness data to the stool bank, but would need not have to be official sub-investigators. And we think such an observational study would give physicians, we know, would give them access to carefully screened stool product, but it would also allow capture of data that is currently not being captured by stool banks. So the study would likely also provide data that is not being obtained from the IND studies currently being conducted as a result of their strict inclusion and exclusion criteria. So that's all I have to say, and I'm happy to answer any questions you might have, or try to. So Christian, are there instructions?
Yeah. So if folks want to ask questions, I think the easiest thing to do is go to the chat box and type them in, and then we can read them and answer them. I mean, if you want to take yourself off mute and ask them aloud, that's fine too. We have about 10 minutes for questions. So feel free to jump in, because, I mean, that was a lot of really good information. So we have one here now from Lisa McGifford. It says, so as of now, drugs in development and everything else is status quo.
Yes. Continuing to allow OpenBiome to perform to sell FMT or provide it for physicians, and they can do the procedure in their office. The drugs are continuing to move through clinical trials. I don't know how long it's going to take them to complete that process, or if they'll get the results that they hope for.
So I would say two things while we're waiting to see if other questions roll in. So Peggy Lillis Foundation doesn't take positions on any treatment. We just try to provide as much information that is publicly available and verified, so that patients and clinicians have all the information they can to make the best decision for each patient. But we've been interested in this primarily because we felt that there was a real dearth of patient participation in this conversation. And the public meeting last November that Professor Hoffmann and I both spoke at was, as far as I know, the first time that a patient advocacy group actually was added to the agenda. And yet that was the fourth public meeting in 7 years. So we know, even from just talking to many of the companies like RebuyOtix and Sirius, who we also regularly partner with, that, you know, they also feel that there's a lack of patient voice in this space. And so, you know, that's another reason why we wanted to do this webinar, was to get people some more information so that they could hopefully take a more active role, you know, in telling the, in telling the FDA what they want, what we need. So we have 2 more questions. Lisa McGifford says, how do patients ask for FMT? Are doctors typically aware of the options?
Yes, I think any gastroenterologist is very much aware of that this is being done. Not all of them do it, but my understanding is that their OpenBiome has a map on their website that indicates that most people are within, I don't know, something like 20 miles. Do you know what it is, Christian? A physician who will.
Yeah, I don't remember it either off the top of my head.
But it's basically, there are physicians across the country who are doing the procedure now and that get their stool from either their hospital stool bank or from OpenBiome.
Which is something that you did mention, Professor Hoffmann, but I think is also worth sort of reiterating, which is there are many academic medical centers. I can think of Mayo Clinic off the top of my head, also Massachusetts General, that are operating their own internal stool banks for both research and FMT treatment purposes. So, you know, in the event that OpenBiome was impacted by them suspending enforcement discretion, there would still be some availability. It's just it would be limited, and it's unclear how we would respond to that at this time.
Unfortunately, there's no regulation of stool banks per se, so we don't have a list of all the hospital stool banks in the country.
Right.
So if they're half a dozen or 2 dozen.
So it's interesting.
It is.
So we have a couple more questions. So there's one, are there any data on the extent of DIY FMT?
No, at one point there was a figure on the FMT Foundation's website that said 10,000 a year, but I don't know how that number was arrived at. And so I think it's certainly hard to get a handle on it, for sure. But I'm sure there have been a few thousand a year. I don't know if it's 10 or more.
Yeah, we, us and the Fecal Transplant Foundation collaborated with a few doctors at Brown University to do surveys of our patient bases. And so there is a study that just came out, I believe, in January, that it's not about the extent of it, but it's about like why did people do it, what have their outcomes been, to the best of the ability of those researchers to suggest. And I will include a link to that when we send a follow-up email for this webinar so people can see that as well. Then we have a question, can you provide an example of a tissue that is regulated as both a drug and a tissue? And I don't know.
I should have had that at the tip of my fingers. No, I could probably find out, though, but off the top of my head, I don't know.
I can. Hi, Dr. Hoffmann. This is Gerard Honig. I'm a member of the board of trustees of the Peggy Lillis Foundation, and I'm a researcher by trade. So, I mean, if you can think of, there are a handful of such products. I can think of one, for example, which is a stem cell derived product. So this is a product which is basically highly processed and cultured fat cells, which are used for topical application for Crohn's disease. So this is a product. It's actually not approved in the FDA currently. It is currently undergoing a phase 3 trial in order to support an eventual FDA registration under the biologics rules. And it has been approved in Europe. So there are a handful of products like this where human cell-derived products are basically put down the drug pathway as opposed to, I mean, even you could think of something like intravenous immunoglobulin would be another example of that versus other products which conceptually seem quite similar, such as hematopoietic stem cell transplant would be going down the human tissues regulatory route. I hope that addresses the question.
And then we have one, oh, hold on a second. Could you provide some further clarity on defining potency in the context of fecal material? What is your view on using viability alone as is currently performed?
Currently. So I should say I'm a law professor, not a scientist. I'm not the best person to ask, but I mean, I think I know both with probiotics and FMT that this is, there is an issue about, how many, what's live in terms of at the point of administration versus the point of recovery, and so I think that's one issue. But I don't know, what are other options that are available for determining potency, and there may be obviously someone else on the call who has some idea for that.
Then, yeah, I don't know the answer to that either. I am also not a scientist, and I know that it's sort of an ongoing challenge to determine potency from these kind of treatments. So given the microbial component of stool is thought to be the primary driver of therapeutic effects, can you think of any reasonable justification for fecal matter being classified as a tissue?
I'm sorry, can you repeat the question?
Yes, sorry, my dog was barking, so I muted myself for a second. So given that the microbial component of stool, basically the microbes, is thought to be the primary driver of any therapeutic effect, can you think of a reasonable justification for fecal matter being classified as a tissue?
Bless you. So again, my colleague Alex Koretz has made that argument. I'm not a scientist or a medical person, so I'm probably not the best person to answer that.
So, sorry. We moved into a new building that has an elevator. Whenever it dings, the dog goes nuts. She's not used to it yet. So if there are no other questions, I think we can call it a day. Sorry for those that we couldn't answer some more of the scientific questions. This was really a policy conversation, but we do appreciate them being asked. If I can find any information about that, I will share it. We're going to send out a recap email with this recording and the other research that I mentioned, so folks have everything. I just really want to thank Professor Hoffmann for taking time out of what I know is a busy schedule, and also for her sort of ongoing advocacy in this space. I think it's really important that we have sort of ethical and normative conversations about treatment, and we look at it from that lens as well as the science lens. So I really appreciate you and other people who bring that to the table.
Thank you.
Well, thank you again for doing it. Thank you all for joining us. We had about 42 people live, so that was, I think, great. And we will be sharing our next topic soon. Thank you again, Professor Hoffmann. We really appreciate it.
You're welcome.
Bye-bye.
Bye.